Imagine getting injured and suddenly needing a blood transfusion. A perfectly healthy person is standing right beside you, ready to help. Sounds simple enough, right? But a doctor cannot just take that person's blood and pump it into your body. One wrong match, and your immune system can react almost immediately, turning a life-saving procedure into a dangerous emergency. Today, we know these differences as blood types A, B, AB, and O. But tens of thousands of years ago, ancient humans knew nothing about them. They hunted, raised children, and migrated across the planet while an invisible biological divide existed inside their bodies. So why can two people belong to the same species, have the same red blood, and still have blood that refuses to mix? The strange part is that red blood cells look almost identical. They are tiny, round, and red. But here's where the real difference hides. Covering the surface of those cells are microscopic sugar molecules, almost like secret molecular crowns. Type A carries one pattern, type B carries another, type AB carries both, while type O lacks the A and B versions. Your immune system inspects these markers like an extremely suspicious security guard. If it encounters the wrong one, it attacks. That may sound like an absurdly dramatic response to a few tiny sugar molecules, but the human immune system has never been famous for staying calm when it thinks something does not belong. For years, researchers thought early humans may have started with a single blood type. Then, as modern humans evolved and spread into new environments, other blood types gradually appeared. It was an appealing idea. One human lineage, one original blood type, followed by new versions emerging over time. But then scientists began reading the genetic sequences buried deep inside our DNA, and the story took a very different turn. Blood type diversity was not a recent invention of modern humans at all. Its roots reach far deeper into evolutionary history. And this is where things get fascinating. Genetic evidence suggests that forms of the A and B blood group system have existed for more than 20 million years, long before the human lineage separated from several other primates. In other words, a modern human and a distant primate relative such as a gibbon can still share similar blood cell surface markers inherited across an enormous span of evolutionary time. Think about that for a second. Empires rose and vanished, continents shifted, climates transformed, entire species disappeared. Yet these tiny molecular signatures survived, which raises a much bigger question. Why would evolution preserve them for so incredibly long? Part of the answer began to emerge when scientists decoded DNA from Neanderthals and Denisovans. For a long time, Neanderthals were sometimes assumed to have had a much simpler blood type profile, including the idea that they were mainly associated with type O. Neanderthals have a long history of being underestimated. Ancient DNA changed that picture. Researchers found genetic variants connected to A, B, and O blood group types among these ancient human populations. Their blood biology was far more diverse than the old stereotype suggested. They were not some crude side branch with a simple biological system. They were complex human populations with an evolutionary history much closer to ours than people once imagined. But blood groups revealed something even bigger. Ancient blood group genes can act almost like arrows pointing back toward ancestral origins. Neanderthals and Denisovans carried certain blood group variants that are still found in African populations today. That suggests their distant ancestors carried part of this diversity with them as ancient human populations moved out of Africa and into Europe and Asia. Picture small groups crossing grasslands, deserts, and frozen mountains, carrying stone tools, animal hides, and genetic variants they had no idea existed. Every migration moved more than people. It carried an invisible biological library across the world. But wait. A tiny piece of that library may still survive in some people today. Researchers have identified a rare variant in the rhesus blood group system linked to Neanderthal ancestry. It is uncommon across much of the modern human population, yet has been reported among some indigenous Australians and people from Papua New Guinea. It is almost like an ancient code fragment preserved inside modern DNA. The broader genetic record shows that modern human ancestors interbred with Neanderthals, and some descendants of those populations later continued toward Oceania. The past never completely disappears. Sometimes it simply becomes microscopic and hides inside a drop of blood. That genetic mixing, however, may not always have been biologically simple. Differences in rhesus blood group systems can create problems during pregnancy when a mother's immune system reacts against blood cell markers carried by the fetus. Researchers have proposed that certain incompatibilities between Neanderthals and modern humans could have increased the risk of pregnancy complications and reduced reproductive success in some pairings. A microscopic difference on the surface of a blood cell may therefore have acted as one small biological barrier between ancient human groups. It would not explain Neanderthal extinction by itself, but it may have been one piece of a much larger and more complicated story. The problem is, if different blood types can create so many complications, why did evolution not simply choose the best one and eliminate the rest? On the surface, that sounds efficient. But evolution does not run a factory designed to produce identical humans. If everyone carried exactly the same blood group markers, then a pathogen that became especially good at exploiting those markers could potentially spread through a population with very little resistance. No variation. No biological escape route. Blood group diversity can work like a form of evolutionary insurance. One group may be more vulnerable to one disease, while another has a better chance of surviving it. Imagine your cells as castles and microbes as thieves trying to break inside. Blood group sugars are like locks mounted on the gates. Over time, some pathogens evolve ways to recognize and attach to particular molecular patterns. If every castle used the same lock, the thief would only need one good key. But in a population with different blood types, the locks vary from person to person. A pathogen that succeeds in one body may struggle in another. And that is why the story of blood types is about far more than transfusions. It is really the story of how our ancestors survived against microscopic enemies they could not see, could not understand, and did not even know existed. Imagine an ancient community living in a region thick with mosquitoes, where malaria quietly moves from family to family. Once the parasite enters the bloodstream, infected red blood cells can stick to healthy ones, forming large clusters that interfere with circulation and may block small blood vessels. But here's where blood type matters. In people with type O blood, those clusters tend to be smaller and less tightly packed. Blood can keep moving more freely, reducing the risk of severe complications and death. In the long battle against malaria, type O became something like an invisible layer of biological armor. But evolution almost never gives anything away for free. The same type O blood that offers an advantage against severe malaria can make people more vulnerable to cholera. Cholera bacteria attack the intestines and release toxins that cause the body to lose water at a frightening speed. People with type O tend to develop more severe symptoms, lose fluids faster, and face a higher risk of dying than people with some other blood types. So the very same blood type can be a hero in one battle and a liability in another. Nature, apparently, has a rather cruel sense of humor, and the scars from those battles can still be seen on the map today. In parts of the Ganges River region in India, where cholera has struck repeatedly throughout history, type O is unusually uncommon. Over many generations, people with blood types A and B may have had a better chance of surviving severe cholera and passing their genes on to their children. So the modern distribution of blood types is not entirely random. It is almost like an invisible record of ancient epidemics, with every shift in gene frequency hinting at who survived long enough to continue the story. But that is only part of the picture. Beyond A, B, AB, and O, people can also be divided into two strangely named groups, secretors and non-secretors. Secretors release blood group-related sugar molecules into saliva, intestinal fluids, and other body secretions. Non-secretors do not. The names sound like rival prehistoric secret societies, but the difference is entirely genetic. And inside the gut, those tiny sugar molecules can become exactly the kind of doorway certain viruses are looking for. Here's where things get strange. Norovirus, one of the viruses responsible for severe vomiting and diarrhea, often needs to attach to specific sugar molecules on cells lining the gut before it can infect them. In secretors, those attachment points are readily available, almost as if the homeowner accidentally left handles on every door for the burglar. Non-secretors, however, lack many of those targets. The virus arrives, searches for somewhere to grab on, and may come up empty-handed. A gene that has lost part of its normal function can end up becoming a defense against infection. It sounds like a clumsy evolutionary solution, but sometimes clumsy works. Even more surprising, this trait is not new. When researchers examined ancient DNA, they found that 3 out of 4 ancient individuals in one study carried genetic variants associated with the non-secretor state. That suggests Neanderthals and other ancient human groups may have faced noroviruses or similar intestinal pathogens tens of thousands of years ago. Think about that for a second. While they were hunting animals, protecting their fires, and avoiding predators, one of their most persistent enemies may have been something far too small to see. But do blood groups only matter when we talk about ancient disease? Not quite. During the COVID-19 pandemic, researchers also reported small differences in risk between blood types. Some studies found that people with type O blood appeared to have a slightly lower risk of severe disease, while people with types A or AB sometimes showed a somewhat higher risk. One proposed explanation involves naturally occurring antibodies and blood group-related markers that may influence how the virus interacts with cells. But this effect is modest. Blood type is not a force field, and the virus does not check your blood type card and politely move on. The more interesting idea is how blood group diversity might protect an entire community. When a virus is produced inside a person with type A blood and then passes to someone with type B or O, it may carry molecular markers acquired from its previous host. Natural antibodies in the next person may recognize those incompatible markers and attack the virus more effectively. In that sense, a population with many blood types can behave like a wall built from different layers. A virus that gets through one layer may struggle with the next. Diversity does not guarantee protection for every individual, but it may help slow transmission across the population. Blood type, however, is not only involved in our battles with microbes. It also affects how quickly blood clots after an injury. People with blood types A, B, and AB generally have higher levels of clotting-related proteins, especially von Willebrand factor, than people with type O blood, often by roughly 30%. When skin is torn, these proteins help platelets gather and form a plug over the wound. Bleeding can stop sooner. In a world without hospitals, stitches, or bags of donor blood, even a small advantage in clotting speed could mean the difference between walking away from an injury and never making it back to camp. But in the modern world, that same advantage can become a weakness. Blood that clots more easily can also form unwanted clots inside vessels even when there is no injury at all. Those clots can block blood flow to the heart or brain, contributing to heart attacks and strokes. That is why people with non-O blood types, including A and B, tend to have a somewhat higher risk of certain cardiovascular problems than people with type O. A trait that may once have saved an injured hunter from bleeding to death can create problems decades later. And that is one of evolution's great paradoxes. What worked beautifully in the past is not always what works best in the present. Put yourself in the place of one of our ancestors tens of thousands of years ago. You are chasing an animal through the forest when you slip on a sharp rock, or a claw suddenly tears through your skin. Back then, the biggest threat was not cholesterol or heart disease decades later. The problem was much more immediate. You could bleed to death within minutes. And this is where blood types A and B may have offered an advantage. With higher levels of certain clotting proteins, wounds could seal more quickly, giving an injured person a better chance of surviving long enough to make it back to shelter. But hunting was not necessarily the most dangerous moment. Picture an ancient mother giving birth inside a dark shelter with no doctor, medicine, or operating room anywhere in sight. If severe bleeding began after delivery, there was very little anyone could do. In that situation, faster clotting could become a lifeline. Women with blood types A, B, or AB generally have higher levels of clotting-related proteins than those with type O, which may have helped bleeding stop sooner. A mother who survived could raise her child, have more children, and pass those genes forward. An invisible biological advantage could quietly shape generations to come. Here's where evolution gets interesting. Evolution does not necessarily favor the trait that helps you live the longest. More often, it favors traits that help you survive long enough to reproduce and pass your genes on. Faster clotting might save a young hunter after an injury or help a mother survive childbirth. A greater risk of cardiovascular problems many decades later. From evolution's perspective, that bill arrives very late. It may sound unfair, but nature does not run a retirement program. traits tend to persist when they helped earlier generations survive through the years that mattered most for reproduction. But if different blood types offer different advantages, why did that diversity nearly disappear in some parts of the world? When Europeans encountered indigenous peoples in the Americas, they found that type O was extraordinarily common, especially in many populations of Central and South America. Types A and B were rare, and AB could be nearly absent in some groups. That raised an obvious question. Did type O somehow provide a special advantage in the Americas? Or had something happened in the past that removed most of the other blood group variants? For years, one explanation seemed to fit. Perhaps devastating epidemics such as smallpox had killed disproportionate numbers of people with types A and B, leaving more type O survivors behind. Considering the catastrophic disease outbreaks that followed European contact, the idea sounded plausible. But there was a problem. When researchers examined ancient DNA from people who lived in the Americas before European arrival, type O was already highly dominant. So the pattern could not have been created solely by those later epidemics. The real explanation had to reach much farther back in time. To find it, we have to travel back roughly 15,000 years, toward the end of the Ice Age. Small human populations from northeastern Asia moved through or around Beringia, the cold region that once connected Asia and North America. These pioneers carried only a fraction of the genetic diversity found in the much larger populations they came from. This is the basic idea behind a population bottleneck and founder effect. Imagine taking just a handful of books from a massive library, carrying them across a continent, and then using those few books to build an entirely new library. In such a small founding population, A and B blood group variants could have been rare or even absent simply by chance. They did not need to be weaker. Genetics can sometimes behave like an enormous roll of the dice. As descendants of those early settlers expanded across North America and eventually into Central and South America, the blood group variants they happened to carry became widespread. Type O rose to extremely high frequencies in many indigenous American populations. A random genetic pattern in a small group of pioneers could eventually shape the blood types of millions of descendants. History has a strange habit of turning tiny accidents into continental patterns. But wait. Type O itself is not a completely new biological design. The most common O allele can be thought of as a loss-of-function version of the ABO gene. A small change in its DNA sequence prevents the enzyme from properly adding the final sugar that creates an A or B antigen. As a result, type O red blood cells carry neither the A nor the B marker. It sounds like a manufacturing defect, but biology is full of useful defects. By lacking certain surface molecules, type O cells can interact differently with some pathogens, sometimes reducing the severity of infection or making particular microbial strategies less effective. And ABO is only the beginning. The human body has dozens of other recognized blood group systems, including Diego, Duffy, and Kell. Each carries its own genetic variations, shaped by population history, disease, and migration. The Duffy system provides one of the clearest examples. Certain Duffy-negative variants reduce the ability of Plasmodium vivax malaria parasites to enter red blood cells. The Diego system, meanwhile, occurs at distinctive frequencies in some Asian and indigenous American populations. Blood groups can therefore act like genetic footprints, helping researchers reconstruct the routes ancient populations followed across the planet. So the next time you see a drop of blood, remember that you are looking at more than a red liquid. You are looking at a living archive. Written across the surface of those tiny cells are traces of malaria, cholera, dangerous childbirth, injuries, migrations, and ancient population bottlenecks. Blood types are not simply random biological clutter. They are the result of millions of years of mutation, trade-offs, disease pressure, and human movement. And perhaps the most remarkable part is this: the survival story of people who lived thousands, even millions, of years before us is still flowing through your body right now.